sir, audible, sir.
Yeah. Good morning to one and all. It gives me great pleasure and honor to be a part of this international webinar organized by the Shramajit Shikshan Prasarak Mandals, other senior college, Umarga, and special thanks to the Department of Chemistry, Industrial Chemistry and Internal Quality Assurance Cell of other senior college, Umarga, have given me an opportunity to share my views regarding the current perspectives in chemical sciences. Being an alumni of this particular college, other senior college, and I feel proud because just my teacher have introduced me, Professor Mullah sir, and a very nice introductory remarks delivered by Honorable Principal Dr. Dilip Garud sir. All my respected teachers from other senior colleges, the faculty members from various parts of the Maharashtra, India, the researchers from India as well as abroad, dear participants, researchers, students from chemical, biological as well as pharmaceutical sciences, and the resource person from USA, Dr. Lahu Chavan, as well as Dr. Shivshankar. Today we have gathered here to discuss recent advances in chemical sciences. In the topic which I will share over you, that is definitely will be the beneficial for the young researchers of not only from the Marathwada region but also Maharashtra as well as India. Before going to talk, just I will share my screen. Please, Mute sir, please allow me to share my presentation. Hello?
Yes, sir. Yes, yes, I am trying.
Yes. Please mute all participants.
Sir, you can.
Yeah. Okay.
Reddy sir and Dindore sir, kindly mute your mic. Reddy sir and Dindore sir, kindly mute your mic.
Sir, slides are visible, sir.
Yes, sir. Yes, sir. It is visible, clearly visible, sir.
Yeah. Okay. The topic which I have chosen is click chemistry. Tiny contribution is nothing but small contribution which we have made in terms of click chemistry. Being an alumni of this particular college, I should give something to my young friends from the college as well as related colleges from the Marathwada as well as from Maharashtra. Before going to talk on this particular aspect, because the participants from various disciplines and my topic is totally multidisciplinary. Hello sir, Mote sir, please mute all. Hello students.
Yes, sir.
Please mute all. Otherwise you can leave.
Yes, sir. I have muted all. You also kindly unmute and then start, sir.
Yes, sir. Okay.
But sir, okay.
The
problem is that if someone unmute again, then we need to mute all every time.
Okay, sir. I can understand. Before going to talk on tiny contribution related to click chemistry, we will focus on the top 10 causes of death in India, because we used to consider this particular international webinar should be multidisciplinary. It should be well connected to biology as well as pharmaceutical sciences. As we know, the different causes of death in India, we all have witnessed about the various patients related to heart disease, chronic pulmonary disease, diarrhea, stroke, lower respiratory infections, as we have witnessed from 2019 to till today regarding this COVID-19 pandemic coronavirus, tuberculosis, neonatal preterm birth, self-harm, as well as a lot of road injuries and other different causes of death in India. Not only this, but also in addition to this COVID-19, that particular virus have made drastic deaths in India, not only in India, but also all over the world. And we all have witnessed about that particular disease or cause of death, not only in India, but also worldwide. I will talk out some of the important causes of death in India, that is tuberculosis. From long back, we have witnessed that in every village of Maharashtra as well as India, we have seen at least 1 or 2 patients in rural places, not only in rural places, but also in metropolitan cities also. Because of this tuberculosis, we know the various kinds of signs and symptoms. If you have some kind of chest pain or some fever, or if you are failing to proper sleep in the nights, or you have some cough, or you are losing lot of weights, these are some of the symptoms of tuberculosis, and these are some primary symptoms. And we know very well, if you are facing a problem of tuberculosis, some other diseases may come with you, like diabetes, like HIV, in addition to TB. And this TB can be transmitted from the person via the droplets of the patients having active TB. Okay, and see, this tuberculosis is one of the top 10 causes of death worldwide. And as per the data published by World Health Organization for the year 2015, more than 10.4 million people fell ill with tuberculosis, and 1.8 million died from the disease. And over 95% of tuberculosis death occur in low and middle income countries. See, the disease, the majority of the patients belongs to the low and middle income countries, and 6 countries account for more than 60% of the total, with India leading the count. Means the majority of the patients from the India, followed by Indonesia, China, Nigeria, Pakistan, and some of the countries from South Africa. Means India is leading in terms of tuberculosis. See, to cure this particular disease, primary tuberculosis as well as secondary tuberculosis, like multi-drug resistance tuberculosis or total drug resistance tuberculosis, or XDR also, there are different types of tuberculosis. And the journey of different kinds of drugs available in the market, and the first drug was discovered in the year of 1943 to cure or to treat tuberculosis was para-amino salicylic acid. It is a very small drug. Even though today also doctors are prescribing this particular drug for the patients of tuberculosis. Then later on, streptomycin, which came in the market and have made a remarkable progress. And in the year of 1946, small molecule again thiacetazone, and in 1952, again very small molecule isoniazid came in the market. And this particular drug have made a drastic change till today, even though after 70 years, doctors are prescribing this first line drug, that is isoniazid. Then in the year of 1952, pyrazinamide, again it is a small drug, and rifampicin in the year of 1957 has totally changed the strategy in the chemotherapy of tuberculosis. Then later on in the year of 1961, again the small drug was developed by the Lederle Laboratories, ethambutol, and then fluoroquinolones and rifampicin have came in the market to cure this tuberculosis. See, later on, the only one drug can survive the patients of tuberculosis, is nothing but bedaquiline, and we will see about that particular drug also. Okay, and see, just I'm showing you over here what are the first line drugs, what are the second line drugs, and newer second line drugs. The first line drugs, again, they are very small in terms of structure, like isoniazid, ethambutol, pyrazinamide, and no doubt streptomycin. Hello? Streptomycin and rifampicin, they are bulkier drugs, somewhat macrocyclic compounds. And second line drugs, that is thiacetazone, paraamino-salicylic acid, kanamycin, these are the second line drugs. And the newer second line drugs is nothing but higher antibiotics, that is ciprofloxacin, ofloxacin, levofloxacin, clarithromycin, azithromycin. See, in COVID-19 pandemic, we have learned one of the term, that is a repurposing of the drug. No doubt the drug were used by the doctors to cure this COVID-19 disease, that is hydroxychloroquine or chloroquine. The purpose of making these two drugs was different, but later on that particular drug having utilized to cure this COVID-19 virus. In addition to that, we all have witnessed that rifampicin. The purpose of making that particular drug rifampicin was different, but the use of rifampicin for the COVID-19 we have seen means the terminology, that is a repurposing of the drug. No doubt, another kind of antiviral drugs, we have witnessed that different types of antiviral drugs, that is at the end of their names, vir will be there. That is acyclovir, favipiravir, such kind of drugs. No doubt they are antiviral drugs, and this COVID virus is also one kind of viral drug. And we have learned that repurposing of the drugs. No doubt I will show you the structures of these anti-tubercular drugs, because whatever chemist, medicinal chemist, they are doing the research related to the structures of the kanamycin, rifampicin.
Sir, kindly unmute your mic and then speak.
Yeah, just I am showing the structures of the drugs. That is the isonicotinic acid hydrozide, very small drug, INH, pyrazinamide, ethambutol, very small drugs. And these are the drugs currently used or prescribed by the doctors. This drug, the last drug, bedaquiline, and experts in the field of tuberculosis, they are suggesting that this particular drug can survive the patients of tuberculosis for next 10 years. Because in recent years, in 21st century, we have seen that some of the patients are not supporting the drugs which are available in the market. That's why doctors are prescribing combination of first line, second line drugs in the form of third line drugs. And this particular drug can be used to cure this particular disease. Then another type of disease is nothing but fungal infections. And in summer or in the winters, we have seen some of the people are facing the problem of infection. It may be bacterial infection or fungal infections. No doubt that tuberculosis is a bacterial infection. This fungal infections, there are two types of fungal infections are there. One is a superficial, and we have seen on the hands or legs or on the eyes of various people, such type of ringworms are available. And this is a superficial. And another type of fungal infections is nothing but systematic fungal infections, and which occurs by the fungi, that is a Candida albicans, and which is a opportunistic fungal infections. And to cure this particular fungal infections, the drugs which are used to cure these fungal infections, a lot of antibiotics doctors are prescribing, that is amphotericin B, azole antifungals are there, that is ketoconazole, itraconazole, fluconazole, etc. Different types of allylamines and pyrimidine based drugs were used to cure the fungal infections. See, as I have told about the azoles, that is fluconazole, itraconazole, miconazole, these azoles are synthetic compounds. They are not naturally occurring compounds. There are two types of azoles. One is imidazole based or triazole based group of antifungal drugs, which inhibits the enzyme cytochrome P450 of 14 alpha D methylase. And this particular enzyme converts lanosterol to ergosterol and is required in fungal cell membrane synthesis. And these drugs also block steroid synthesis in humans. As I told you regarding two types of azole antifungals, one is imidazole based antifungals like miconazole, bifoconazole, ketoconazole, etc. And triazoles, again, there are two types of triazoles we will discuss. These triazoles, they are newer, less toxic, and more effective. The examples are fluconazole, rauconazole, posaconazole, itraconazole, voriconazole. And I will show you the structures of those triazoles and see the arrangement of the nitrogens in triazoles. They are at 1 to 4 position. In fluconazole, in voriconazole, 1 to 4 position is there. In itraconazole, again at 1 to 4 positions. And there are many more examples of triazole based antifungal agents. Now, we all have witnessed in the month of October. On 5th October, the Nobel Prize for Chemistry was declared to the three great personalities from the chemistry background, and for their development of click chemistry and bioorthogonal chemistry. That Nobel Prize was given to Professor Caroline Bertozzi from Stanford University, USA, Professor Meldal from University of Copenhagen, Denmark, and Professor K. Barry Sharpless from Scripps Research Institute, USA. These three people have made a lot of contributions in terms of click chemistry. And we will talk about what do you mean by click chemistry. See, this click chemistry, as I told you that these three people, Bertozzi, Meldal, and Sharpless, for their development of click chemistry and bioorthogonal chemistry, means it is the application of chemist made by Bertozzi, and Bertozzi have done the click chemistry approach on biological systems. That's why first Sharpless have coined this terminology click chemistry in the year of 2001, and first time he has reported the terminology click chemistry with a chemical structure called as a triazole. And later on, in the year of 2002-2003, Professor Bertozzi again took the next step of developing click reaction that could work inside a living system, that is a biological system. That's why it is called as bioorthogonal reactions or bioorthogonal chemistry. What is this click reaction? Just in short, I will tell you, it is nothing but reaction between terminal alkyne and terminal azide in presence of copper catalyst, and which gives corresponding 1,4 disubstituted 1,2,3 triazoles. And we have seen in the last month, most of the chemistry people, they have kept their profile of this particular page. And sir, lot of disturbances are there. As I told you, this is nothing but one kind of cycloaddition reaction between terminal alkyne and terminal azide in presence of copper catalyst, which leads to regioselectively 1,4 disubstituted 1,2,3 triazoles. This click reaction discovered by parallelly Sharpless as well as Meldal in the year of 2001. This both of them have independently carried out this particular reaction. And this particular reaction is nothing but one kind of cycloaddition reaction which gives 100% atom economy. See, I will show you over here in terms of some of the images. This is nothing but azide, terminal azide, and this is the alkyne in presence of copper catalyst, which leads to 1,4 disubstituted 1,2,3 triazoles. And the same approach have taken by Professor Bertozzi in terms of biological system, and they have taken some kind of cycloalkyne. See, here cycloalkyne along with the the azide, and this cycloalkyne from the living system, and without use of any metals, means copper catalyst, they have performed, Professor Bapurao have performed this particular reaction into biological system, leading towards the 1,4-disubstituted 1,2,3 triazose. See, in short, I will show you once again. This is the azide and this is the alkyne, terminal alkyne. When they come to each other in presence of copper catalyst and formation of carbon nitrogen, bond takes place, and when they come to each other, it is nothing but some kind of click type of reaction takes place, means like a voice like this one. Okay, see, the great personality K.P. Sharpless have received a second Nobel Prize for his contribution, and he is the only fifth person to win the 2 Nobel Prizes. Earlier to this particular, in the year of 2001, he has received another Nobel Prize for his great contribution in asymmetric oxidation reaction, and the researchers, faculty members, students of postgraduate, they have studied Sharpless asymmetric epoxidation reaction, Sharpless asymmetric dihydroxylation reaction, Sharpless asymmetric amino hydroxylation reaction. Means for asymmetric oxidation reaction, he has received a first Nobel Prize, and the second Nobel Prize he has received for his contribution in terms of click chemistry. Okay, see, I will tell you how this reaction takes place, how this click chemistry reaction takes place, or click reaction. As I already mentioned, the terminal azide and the terminal alkyne in presence of copper catalyst gives to 1,4. This is a 1,2,3,4, 1,4-disubstituted 1,2,3 triazose. This click chemistry is a simple name for a simple set of reactions. The core of the click chemistry concept is the cultivation of small set of reactions that are extremely ideal for working with organic molecules. And Sharpless described them in his famous 2001 M.U.Aurangabad paper. See, this click reaction, it is a modular and be wide in scope, means whatever functional groups are available in organic compounds, they remain as it is in the product. Means this reaction condition does not affect on any kind of other functional groups. It gives very high yields. Means, as I told you, 100% atom economy is there, and it generates only inoffensive byproducts easily removed. It can be removed by means of only crystallization, and it is highly stereospecific, highly regioselective as well, requires simple reaction conditions, and you will see later on how this particular reaction can work very well, and require readily available reagents and starting materials. Required no or benign solvent or easily removable solvents like tertiary butanol water itself, or polyethylene glycol, or THF water solvent system. This type of solvent system can be used and it can be removed easily. Requires simple product isolation and purification. No column chromatography, no flash column chromatography is required, and it uses products that are stable in physiological condition, and it is 100% atom economy. Okay. See, why Professor Bapurao, Sharpless and Meldon have received the Nobel Prize. See the history, means the number of publications in terms of year-wise. The first publication came in the year of 2001, and see, in the 20 years only of this click chemistry approach, in the year of 2010, the number of publications was 753. In the year of 2020, the number of publications was 1,371. And for the year, again, the year has not ended yet. The number of publications are 1,081, sorry, 1,081. And see, the total number of publications in the last 20 years, around 17,000 publications. Means every year, you can see minimum 700 to 800 papers are there on this particular concept. And see, fortunately, and I will say proudly, we have started working on this particular concept from 2010. First paper we have published in the year of 2010. And see, we are the followers. We are not inventors, just we are the followers of this particular small concept in organic chemistry. In organic chemistry, that is a click chemistry. And see, as I told you that the 1,4 triazoles are in the market as a fungal, antifungal agents. However, there are several potential pharmaceuticals available in the market having 1, 2, 3 triazole system over here. See the drug that is a CAI having 1, 2, 3 triazole, as well as a TSAO having 1, 2, 3 triazole system. Tazobactam, no doubt it is an antibiotic. Tazobactam, that is beta-lactamase inhibitors. 1, 2, 3 triazole system is there, and cephalosporin or ceftriaxone again having a 1, 2, 3 triazoles, and many more small molecules are under clinical trials of phase 1, 2, 3 as well. With this, we got inspired to make some of these small molecules by considering the starting materials should be commercially available or it should be readily available at postgraduate laboratory, and it can be made easily. Accordingly, we have designed the molecules, and before going to talk on why these 1, 2, 3 triazoles are important in terms of medicinal chemistry, in terms of material science, in terms of polymer science, lot of research were performed in last 20 years, not only in terms of medicinal chemistry, in material science, in polymer science, in biological sciences as well. See, these 1, 2, 3 triazoles containing compounds are showing different types of biological activities like anti-cancer, anti-HIV, anti-inflammatory, anti-hepatitis B, anti-diabetic, antibacterial, anti-tubercular, antifungal, Alzheimer type of properties. Such kind of triazoles were displayed. With this, we got inspired to work on this particular click chemistry. That's why I have given here the title "Tiny Contribution Towards the Click Chemistry." This is a small contribution, and so far we could able to publish around 35 papers in the journals of international reports on this particular small concept that is a click chemistry. And accordingly, we have designed the molecules. Simple molecules are there, and which we can prepare at any places. See, this is a 1, 2, 3 triazole, and this triazole's backbone displays drug-like properties, and this is the aromatic part, and this is a lipophilic control, as well as there is some ethereal linkage. And how to make this particular compound? We have designed this particular molecule and docked against the enzyme like a DPRE1, which is a anti-tubercular enzyme. Okay, we have designed such type of molecules and synthesized from the commercially available starting materials, that is corresponding phenols. See, phenol, small molecule is there, phenol, which is available in postgraduate laboratory. This phenolic OH group gets alkylated by using base and propargyl bromide, this alkyne, because we need a terminal alkyne. It gives a corresponding alkylated products, and these are the some kind of 7 derivatives we have made. And later on, we need a terminal azide, and these terminal azides were prepared from commercially available benzaldehydes. This benzaldehyde aldehyde group on reduction by using sodium borohydride, that is a mild reaction condition, and this alcoholic OH group means benzyl alcohol on reaction with phosphorus tribromide PBr3, which gives a benzyl bromides, and these benzyl bromides on reaction with sodium azide via nucleophilic substitution reaction leads to the corresponding benzyl azides. Now see the things, we have a 7 terminal alkynes, we have a 5 benzyl azides, means we can make 35 different types of 1,4-disubstituted 1, 2, 3 triazoles by using click chemistry approach, and see the reaction conditions over here. As see, this terminal alkynes and this terminal azides, benzyl azides, under click chemistry approach by using copper sulfate in tertiary butanol water solvent system at room temperature for 19 to 28 hours at room temperature. Means just only stirring. It gives a corresponding 1,4-disubstituted, means 1, 2, 3, 4. 1,4-disubstituted 1, 2, 3 triazoles having ethereal linkage. See the structures of all these compounds over here, having substitution on phenolic phenol side as well as substitution on this benzyl azide side. These are the different kinds of substitution on benzyl azide side, that is chloro, nitro group is there. On phenolic side, nitrochloro is there, as well as iodo, and on benzyl side, benzyl azide side, bromo group is also there. After synthesizing these particular compounds, we have well characterized by means of spectroscopic techniques like IR, H1NMR, C13NMR, as well as HRMS spectrometry. And after confirmation of the structures of all these compounds, we have screened these compounds against the tuberculosis strain, that is MTB S37RA, as well as we have screened these compounds for their antioxidant activity by using DPPH, radical scavenging activity. We have determined the MIC as well as IC50 for an anti-tubercular activity, as well as IC50 value for antioxidant activity. And see, some of the synthesized compounds like 22A compound is showing lower IC50 value against the TB strain, that is MTB S37RA. The compound like 23C, 23E having 0.5 ml, 0.5 microgram per ml, 0.7 microgram per ml, as well as compound 26E, 0.9, 0.8, 0.2 microgram, 0.8 microgram IC50 values. And we have used the standard drug, that is a rifampicin. And for screening the antioxidant activity, we have used the standard drug, that is butylated hydroxytoluene, which is commercially available, as well as ascorbic acid. And we got some excellent results for anti-tubercular activity as well as antioxidant activity. As the practical results, the experimental results, as I said that we have talked this particular most active compound, that is a 23E, against the active site of DPRE1 enzyme, and we got some excellent results, excellent interactions between the ligand, that is particular compound, with different types of amino acids through hydrogen bonding. And we got an excellent docking score for these most active compounds. See, in summary, these are the most active compounds with lower MIC value as well as IC50 value for antioxidant activity. All the synthesized compounds we have screened against the for toxicity, and none of the compound is showing toxicity against the cell lyse, that is a THP1A549 as well as a PANC1 cell lyse. In addition to this, we have analyzed through in silico computational study, that is ADME properties, and they are showing better properties. And this particular work we have published in the year of 2015 in the journal MedChemCom, Medicinal Chemistry Communications. In our first attempt, we have undertaken the starting material, that is a phenol, simple phenols. But in our second attempt, see, just we have modified in terms of benzyl azides. We have changed the benzyl azides in place of benzyl azides, amide based azides, and we have designed such type of molecules, and this part we have kept constant. And to see the effect of introduction of this amide group, in between the azide, the triazole as well as the methionine group over there. And accordingly, we have a design, as we have seen the synthesis of this terminal alkyne from corresponding phenols. And here, the aromatic amines, means different types of anilines, were subjected with chloroacetyl chloride, followed by nucleophilic substitution with azide, sodium azide, which gives a corresponding azidoacetamides. See, these azidoacetamides, just we have changed the azide part, and we have kept this particular alkyne part constant, and to see the effect on the different types of properties, therapeutic properties on this structure. Accordingly, we have optimized the reaction conditions by using copper catalyst, different copper catalyst we have optimized, and we got excellent results by using copper acetate in presence of just we have changed the solvent over here in presence of ionic liquid. And after optimization of the reaction conditions, we have synthesized this amide based 1,4-disubstituted 1,2,3 triazoles, and these are the structures of all the triazoles. And after synthesizing, we have characterized very well by means of spectroscopic techniques and screen for their antifungal, anti-tubercular as well as antioxidant activity. Out of synthesized compounds, we got some excellent results for antifungal activity against the 5 strains of fungi, and some of the compounds are showing equivalent activity or twofold activity against the some of the fungal strains, as well as some of the synthesized compounds are showing better antioxidant activity against the standard drug, that is the butylated hydroxytoluene. However, none of the synthesized compound is showing better anti-tubercular activity. But however, this synthesized compound is showing excellent antifungal activity. And the practical experimental results, which we have seen through theoretical results in terms of molecular talking study, and we have talked the molecules against the fungal enzyme, that is a strain, active sites of Candida albicans of lanosterol 14 alpha demethylase, and showing excellent interactions between the different types of amino acids with ligand, that is a compound. Okay, this particular results we have published in the Journal of RSC Advances. In our first attempt, just we have changed azides and we have kept alkyne part constant. In our next attempt, we have kept that azide part, that is benzyl azides, and we have introduced some kind of acyl group, that is a acetophenone type of moiety over here is there. And now we have free this ketone group. We have this free ketone group, and we can expand in terms of billions of the compounds. And we have designed such type of molecules and docked against the sterol alpha 14 demethylase, which is a antifungal enzyme. Okay, and accordingly we have synthesized the compounds from commercially available phenols, which on acylation followed by Fry's rearrangement, which leads to the orthohydroxyacetophenone. And one of the orthohydroxyacetophenone is commercially available, and just we have made 2 orthohydroxyacetophenone by using Fry's rearrangement. And whatever project we are giving to the students of MSc Fry's rearrangement for MSc practicals, just we have collected those samples and we have purified and used for making different types of terminal alkynes by using propargyl bromide in presence of potassium carbonate, DMF, which gives these 3 types of terminal alkynes, and with the help of already synthesized benzyl azides under click chemistry condition, that is a copper acetate in tertiary butanol water solvent system, leads to the corresponding 1,4-disubstituted 1,2,3 triazoles having this acetone, this acyl group intact. And we have made a total 18 compounds, and these are the structures of all the 18 compounds having different types of substitution on acetophenone part as well as the benzyl azide part. And after characterizing by means of spectroscopic techniques, we have screened these particular compounds for their antifungal activity against the different types of strains, like 5 strains, as well as we have screened these compounds for antioxidant activity by using DPPH radical scavenging assay. See, among these synthesized compounds, some of the compounds are showing equivalent potency against the standard drug, as well as some of the compounds are showing twofold potency in terms of different types of fungal strains. However, some of the compounds are showing equivalent antioxidant activity, and we have docked these compounds against the active site of sterol 14 alpha demethylase and showing better interactions with the different types of amino acids over here. And in short, these are the most active compounds with lower MIC value as well as IC50 value for antioxidant activity. And we have free this acyl group, means we can make n number of billions of compounds with the help of this particular. And this work is published in the Journal of Chinese Chemical Letters. And when this article was published, the impact factor was around 2, but now the impact factor is more than 9. In our next attempt, just we have changed phenol. In place of direct phenols or orthohydroxyacetophenone, we have changed different types of coumarins over here. Here coumarins are there, and we have designed this type of particular compound, 3 different types of coumarins we have undertaken. And remaining benzyl azide part we have kept constant over here. See, in our first attempt, this resorphenol on pitchman condensation by using ethyl acetate, and this practical we are giving regularly to the students of MSc, and we have collected the samples which prepared by MSc students, and around 1 kg sample we have, and just we have done the purification 2 times, 3 times, and we got this 7-hydroxy-4-methyl coumarin. Now we have a starting material, phenolic OH group is there. This OH group on alkylation by using propargyl bromide, and we have already synthesized the benzyl azides, different types of benzyl azides, which on click reaction by using copper acetate tertiary or water solvent system, which leads to the corresponding coumarin incorporated 1,4-disubstituted 1,2,3-triazoles. Means here we have incorporated 2 different pharmacophoric groups. One is a coumarin and another one is a triazole in a single molecular motif by using molecular hybridization approach. No doubt coumarin itself is active, triazole itself is active, but combination of these 2 pharmacophoric group in a single molecular motif which may give better activities or better therapeutic properties. Accordingly, we have made another one alkyne from commercially available 4-hydroxy-coumarin. Similarly, we have made this terminal alkyne, and these azides were subjected for click chemistry approach, gives corresponding 5-1,4-disubstituted 1,2,3-triazoles, and these are the structures of all the coumarin based 1,2,3-triazoles, characterized by means of spectroscopic techniques and screen for their anti-tubercular activity against MTB S37 RA strain, antioxidant activity, as well as antibacterial activity against the gram-positive bacteria as well as gram-negative bacteria, as well as antifungal activity. And see the data. Some of the compounds are showing better anti-tubercular activity as compared to the standard drug like pyrazinamide. See, pyrazinamide is showing 10 microgram per ml, but our compounds are showing not only 2-fold but 5-fold more active than pyrazinamide. However, our compounds are showing less potency as compared to the standard drug, that is isoniazid. Then in terms of antioxidant activity, our compounds are synthesized compounds are showing equivalent potency for antioxidant activity. However, all of these synthesized compounds are showing antibacterial activity as well as antifungal activity because they are possessing lower MIC values as compared to the standard drug like kanamycin, chloramphenicol, miconazole, ampicillin, like that way. And as these synthesized compounds are showing good anti-tubercular activity, we have docked the molecules against the enzyme that is DPRE1 and showing better interactions with different types of amino acids. And this is the most active compounds in terms of lower MIC value as well as IC50 MIC value for anti-TB as well as antimicrobial. And this particular work, which we have published in the Journal of Medicinal Chemistry Research. In our next attempt, why not to have some different types of coumarin? Again, we have undertaken this particular project from commercially available resorcinol. This resorcinol on Wilson-Huck formulation reaction led to the hydroxy-based salicylaldehyde. No doubt, this is again commercially available. And this salicylaldehyde, which on reaction with diethylmalonate via novel condensation reaction followed by cyclization leads to the another hydrox means phenolic OH based coumarin compound, and this hydroxylated group is subjected for alkylation by using propargyl bromide. Under basic condition, we have only one alkyne, and we have already synthesized the benzyl azides on click reaction by using copper acetate in tertiary butanol water at room temperature leads to the coumarin based 1,2,3-triazoles. And see, these are the structures of all the triazoles, characterized very well by means of spectroscopic techniques and screen for their antifungal as well as antioxidant activity and showing better antifungal activity. That's why we have docked these particular compounds against the active sites of sterol 14 alpha demethylase, and ligands are showing better interactions with the different types of enzymes or amino acids. And these are the most active compounds with a lower MIC value, and this particular work is published in the Journal of Chinese Chemical Letters. Means just we have modified the coumarin part, means 2 different types of coumarins, and we have kept the azide part constant. And in our next attempt, see over here, we have kept coumarin part constant and just we have changed the azide part, and here we have introduced amide-based terminal azides are required to make such type of compounds. Accordingly, we have a design and we know very well how to make coumarin-based alkynes, and we already synthesized this amide-based azides, which on reaction with these two terminal alkynes under click chemistry condition by using copper acetate in tertiary butanol water solvent system leads to the corresponding amide-based 1,2,3 triazoles having coumarin is the one of the important pharmacophoric group in their structures, and these are the structures of two pharmacophoric groups which we have incorporated in the single molecular motif, and after characterization by means of spectroscopic techniques, we have screened these compounds for antifungal activity as well as antitubercular as well as antioxidant activity, and see the red data showing excellent antifungal activity against the different types of fungal strains, and which we have docked against the active sites of cytochrome P450, lanosterol 14 alpha demethylase, and showing better interactions. And this particular work is published in the journals of ArchPharm. Here we have seen phenolic OH group where alkylated and different two types of azides we have used for click chemistry approach. In our next attempt, just we have changed in place of phenolic OH group. We have undertaken one of the backbone, that is isatin backbone over here, and this isatin you will see in different types of drugs. And there is a free NH group, and why not to alkylate by means of propargyl bromide and followed by click chemistry approach. We have designed this particular molecules. And docked against the cytochrome P450, lanosterol 14 alpha demethylase of Candida albicans, and where synthesized from the commercially available this isatin molecule, NH free group is there, which on reaction with a base followed by propargyl bromide, which uses this particular terminal alkyne, and we have already synthesized the benzyl azides from benzyl bromides via nucleophilic substitution reaction, which on click chemistry reaction, click reaction by using copper acetate leads to the corresponding isatin based 1,2,3 triazoles. And here you can see this carbonyl group is a free. You can expand this carbonyl group. You can introduce different types of functional groups over here. And similarly, this alkyne based isatin, which on reaction with aryl azides. These aryl azides were prepared from corresponding arylenes via diazotization followed by substitution with sodium azide, which gives corresponding 1,4-disubstituted 1,2,3 triazoles having this isatin moiety in the final structure. These are the structures of the molecules characterized very well and screened for their antitubercular activity, antioxidant as well as antifungal activity, and see some of the compounds are showing lower IC90 value, lower IC50 value as well as lower MIC value for fungal strains against the standard drugs, that is rifampicin, isoniazid, butylated hydroxychloroquine, miconazole, fluconazole. And after studying the molecular docking study, they are showing better interactions with the cytochrome P450, lanosterol 14 alpha demethylase of Candida albicans. And this particular work, which we have published in the Journal of Heterocyclic Chemistry, and these are the most active compounds with lower MIC value. And none of the synthesized compound is toxic against the cell lines, that is HeLa A549 as well as PAN-1 cell lines. And we also studied their ADME properties. Here we have made the alkyne from isatin. In our next attempt, see, we have changed the one backbone, that is a benzthiazolone backbone, and having this NHN, and we have designed this particular compounds because this benzthiazolone shows available in various kinds of drugs, and we know very well how triazole displays various kinds of biological properties, and accordingly we have synthesized from the commercially available orthoaminothiophenols, which on reaction with chloroacetic acid followed by reaction with sodium hydride base and propargyl bromide leads to the this terminal alkyne having benzthiazolone skeleton. We have only one alkyne. We already synthesized the benzyl azides as well as aryl azides unsubjected with copper acetate in presence of tertiary butanol water solvent system under click chemistry condition at room temperature leads to the benzthiazolone based 1,4-disubstituted 1,2,3 triazoles, and these are the two types of compounds which we have made, and these are the structures of all the benzthiazolone based 1,2,3 triazoles characterized very well by use of H1 NMR, C13 HRMS as well as IER, and screen these compounds for their anti-tubercular activity as well as antioxidant activity, and some of the compounds are showing excellent anti-tubercular activity as well as anti-tubercular activity. As these synthesized compounds are showing anti-tubercular activity, that's why we have doctored these particular molecules against the active sites of DPRE1 enzyme and showing better interactions with different types of amino acids or proteins, you can call it. And these are the most active compounds with a lower MIC value, and none of the synthesized compounds is toxic against these three types of cell lines. And this particular work, which we have published in the Journal of Bioorganic and Medicinal Chemistry, and in this particular structure, you have a free this methylene group you can utilize for many organic transformations as well. Okay, in our next attempt, see here, we have expanded our contributions in terms of click chemistry through base-thiazinone based compounds. In our next attempt, this thiazolidinone, core is available in various kinds of anti-diabetic drugs. And if this NH group is free, we can make its corresponding terminal alkyne, and then why not to have a click approach through different types of benzyl azides by using copper catalyst. And as we know, as this thiazolidinone displays various kinds of anti-diabetic activity as well as anti-inflammatory activity. We have designed such type of molecules and synthesized from commercially available three types of aldehydes, chlorobenzaldehyde, fluorobenzaldehyde, or simple benzaldehyde, which on reaction with this thiazolidinone under Navier condensation reaction by using sodium acetate resulted into the arylidine thiazolidinone. Now we have free NH group, which on reaction with a base like sodium hydride, propargyl bromide leads to the terminal alkyne, and this thiazolidinone based terminal alkyne, which on click reaction by using copper acetate with benzyl azides leads to the corresponding arylidine thiazolidinone based 1,4-disubstituted 1,2,3-triazoles. And see here, we have incorporated two pharmacophoric groups, active pharmacophoric groups, that is thiazolone as well as a triazole in single molecular motif. And these are the structures of all these, and characterized very well and screened for their anti-inflammatory activity, antioxidant, anti-tubercular against the two different strains of TB, that is MTBS37RA, MBOS BCG. And see over here, for anti-inflammatory activity, the standard drug is a diclofenac sodium, which is showing 0.722 value. But however, our synthesized many compounds are showing lower value like 0.70, 0.85, 0.84, means here 26E compound is showing lower IC50 value, lower value, in terms of the standard drug, that is a diclofenac sodium. However, our synthesized compounds, none of the synthesized compound is showing anti-tubercular activity against these two strains. Okay, means these are the somewhat negative results, but they are showing better anti-inflammatory activity. See, these synthesized compounds we have screened for their antibacterial activity against gram-positive, gram-negative bacteria, as well as antibacterial, antifungal activity against the standard drugs. That is 3 antibacterial drugs and 3 antifungal drugs. And our synthesized compounds are showing, some of the compounds are showing 2-fold activity or 4-fold activity, means how they are selective. They are not showing any kind of tubercular activity, but however, they are showing antibacterial as well as antifungal activity. As these synthesized compounds are showing anti-inflammatory activity, that's why we have doctored these particular compounds against the means PPA or gamma, that is the active sites of human peroxisome proliferative activated receptor gamma, and it gives better interactions. And these are the most active compounds for anti-inflammatory activity. Some of the compounds are showing better antimicrobial, that is antifungal as well as antibacterial, and some of the compounds are showing antioxidant activity. And here, this particular work which we have published in Indian patent, as well as we have published some part of work in one of the journal, that is polycyclic aromatic compounds, and the antimicrobial part which we have communicated to some of the journal, and very soon we will receive excellent results for this particular work. Means on single work, 1 patent and 2 publications definitely. In our next attempt, why not to use some different strategy to make 1,4-disubstituted 1,2,3-triazoles, and as we have seen how INH, that is isonicotinonic acid hydrazide, acting as a better anti-tubercular drug, and why not to incorporate this triazole moiety on one of the structure, as well as why not to have some hydrazone type of moiety over here. Accordingly, we have designed the molecules and synthesized them very well from the commercially available para-hydroxy benzaldehyde, which out reaction with the propargyl bromide under basic condition resulted into the alkylated benzaldehyde, and this alkylated benzaldehyde out reaction with the benzyl azides under click chemistry approach resulted into the 1,4-disubstituted 1,2,3-triazoles having this aldehyde group free, and this aldehydes, triazole based aldehydes, were subjected for condensation reaction with different types of acid hydrazides like the isonicotinonic acid hydrazide, which is a drug directly, nicotinic acid hydrazide, as well as para-nitrobenzoic acid hydrazide in methanol by using catalytic amount of acetic acid resulted into the corresponding hydrazoles. And these are the structures of all those hydrazoles, characterized very well and screened for their anti-tubercular activity. And see here, better structure activity relationship you can observe over here. These red marked compounds are having INH, that is isonicotinic acid hydrazide. How that particular drug is showing, how that drug is used to cure TB, and we have practically observed that INH based compounds are showing excellent anti-tubercular activity as compared to nicotinic acid hydrazide or that benzoic acid hydrazide. These two compounds are not showing any kind of anti-tubercular activity. Only INH based compounds are showing anti-tubercular activity as well as antioxidant activity. See over here. And we have talked about this particular compounds against the DPRE1 enzyme and showing better interactions. And these are the most active compounds with a lower MIC as well as IC50 value against the two different strains of TB, that is MTB S37RA as well as M BCG strain. Okay, and these synthesized compounds are not showing any kind of toxicity against these three cell lines and analyzed for ADME properties. And this particular work is published in the Journal of Mini Reviews in Medicinal Chemistry. In our next attempt, in place of para-hydroxybenzaldehyde, we have taken orthohydroxybenzaldehyde, and the same strategy we have used to make alkylation, and followed by click chemistry approach. We have this aldehyde part, and this aldehyde group we have subjected for condensation reaction by using different types of acid hydrazides, that is INH, nicotinic, para-nitrobenzoic, para-chloro, para-methoxy, benzoic acid hydrazide under similar type of reaction condition over here. In place of para-hydroxy, here we have taken orthohydroxy, and these are the structures of all the compounds. 40 compounds are there, and characterized very well by use of spectroscopic techniques and screen for anti-tubercular activity against the two different strains of MTB S37RA, that is a dormant as well as a active strain of TB as well as antioxidant activity. And see here 24A, this 24F, 24K, 24P as well as this 25M, 25H, this 25E and 25A. These are the compounds are having in their structure, that is INH based hydrazoles are there, and they are showing equipotent activity as compared to the standard drug, that is INH, rifampicin, and we could make able to have this kind of structure activity relationship over here as the most active compounds with lower MIC or IC50 value compounds. These are the most active compounds are showing lower MIC value, and none of the synthesized compound is toxic against the four cell lines, and also studied ADME properties. And this particular work, huge work is there, which we have communicated to some of the excellent journal of medicinal chemistry. In our next attempt, we have introduced another pharmacophoric group in the final structure of triazoles, that is a tetrazolopyridine. Pyrroline we can see in many drugs, and why not to incorporate triazole over tetrazolocurulin type of moiety. We have designed such type of molecules and synthesized from commercially available. That is a meta-anicillin. You can change this anicillin or toluidine. You can take this meta-anicillin on acylation, gives acetaldehyde, which on Wilsmer formulation reaction gives corresponding chlorocurulin based aldehyde, that is 2-chloro-3-formylquinone, which on reaction with sodium azide resulted into the tetrazolocurulin aldehyde. Reduction of this aldehyde group OH followed by methylation and subjected for nucleophilic substitution reaction with sodium azide. Now we have only one azide having tetrazolocurulin skeleton, and we have already synthesized different types of terminal alkyls from phenols subjected for a click chemistry approach, resulted into the corresponding tetrazolocurulin based 1,4-disubstituted 1,2,3-triazose. And here we have undertaken one of the examples, that is a phenylacetylene, to compare the biological activity of having this type of azide as well as this type of terminal alkynes. And these are the structures and screen for their antifungal as well as antibacterial activity. And see over here 20 compound, which is a starting material, never shows any kind of bacterial as well as a fungal activity. However, our synthesized compounds are showing two equivalent or four-fold activity as compared to the standard drug in terms of antifungal as well as antibacterial activity, and docked against the sterol 14-alpha demethylase and showing better interactions. And these are the most active compounds. And this particular work, which we have published in the journals of polycyclic aromatic compounds recently. So far, we have seen how to make just a part of our research contribution in terms of click chemistry. I have shown, and whatever work I have shown over here, I have not extended further study over here. Just I want to show quickly how to make a dimeric 1,2,3-triazose. So far, we have seen how to make a single triazose, monomeric triazose, by using click chemistry approach. And here we have designed dimeric 1,2,3-triazose via click chemistry approach, and to maintain the symmetry like human body, like this one. How to make a dimeric triazose over here by using some spacer, or you can call it a linker, and accordingly we have designed the molecules and synthesized from commercially available aromatic amines, as we have seen how to make this type of azides from corresponding anions, which on chloroacylation followed by nucleophilic substitution with sodium azides. We have these kinds of amide-based azides, and we have commercially available resorcinol or catechol under similar type of reaction condition, basic condition, that is potassium carbonate, propargyl bromide, which gives a dialkyne over here, and here also dialkyne. Two dialkynes, and we have a n number of azides on click chemistry approach by using copper sulfate sodium ascorbate in pH 400 water solid system at room temperature leads to the corresponding amide based dimeric 1,2,3-triazose derived from resorcinol as well as catechols. And these are the structures of all the synthesized compounds, well characterized by means of spectroscopic techniques and screened for their different types of biological activities. See, these are dimeric triazoles were screened for their antitubercular activity, antifungal activity, as well as antioxidant activity. In addition to these, some of the compounds were screened for their anti-cancer activity, that is anti-proliferative activity against the 3 cell lines, that is MCF7, A549, and other cell line also. And we also studied percentage EG for the inhibition of against the cell line, that is MCF7, and some of the compounds are showing excellent percentage of EGFR, as well as our synthesizer, one of the synthesizer compound is showing excellent anti-proliferative activity against the standard drug. However, almost 50% synthesizer compounds are showing better antioxidant activity, and most of the compounds are showing excellent antifungal activity, and 2 compounds are showing anti-tubercular activity. See, accordingly, this is the structure activity relationship part, or the most active compounds for anti-proliferative, anti-TB, antifungal, as well as we have also performed their enzymatic assay against the lanosterol 14-alpha demethylase inhibition as well as antioxidant activity. Similarly, we have docked against the cytochrome P450, as well as these synthesized compounds are showing better anti-proliferative activity. That's why we are docked against the EGFR enzyme, as well as we are also docked against the antioxidant M.P.O. enzyme. And see over here, on this particular study, we have published 3 total papers. One is on anti-proliferative activity, one is on antifungal activity, and another is on antioxidant activity on the same compounds. Okay, see, in our next attempt, just we have kept that alkyne part constant and we have changed the azide part over here. That is a simple azides based on aliphatic system secondary aliphatic amines, which we have used, and accordingly we have made azides. And we have already synthesized this type of dialkyls on click reaction by using copper sulfate resulted into the dimeric triazoles, and these are the structures of dimeric triazoles. And these particular compounds we are screened for their anti-tubercular activity as well as antifungal as well as antioxidant activity, and the bold data showing excellent TB, antifungal as well as antioxidant activity. And we are talking against the fungal strain of sterol 14-alpha demethylase, and this particular work is published in the excellent journal having impact. See, in our next attempt, just we have changed the spacer in place of phenolic OH group. We have inserted it is a piperazine moiety, and it looks like one kind of symmetry over here. And accordingly, we have designed the molecules that piperazine based compounds are highly active, and in one of the drug that is rifampicin having a piperazine skeleton. And accordingly, we have made the dialkyne part from commercially available piperazine by using sodium hydride propargyl bromide. We have this terminal alkyne, and we have already synthesized the azide part on click reaction leading to the dimeric triazoles, and these are the structures of dimeric triazoles, and screen for their anti-tubercular activity, antifungal activity, as well as antioxidant activity. And the bold data, bold figures showing excellent tubercular activity, excellent fungal activity, as well as excellent antioxidant activity. And these are the some of the most active compounds. Sorry, these are the some most active compounds in terms of TB, in terms of fungal strain, in terms of antioxidant activity. And we are talking against the fungal, that is, if the synthesized compounds are showing anti-tubercular activity, that's why we are talking against the INHA enzyme and showing better interactions. In short, so far we have seen a part of click chemistry, our small contribution in terms of click chemistry. As I have told you that just we are the followers, we are not the inventors. Similarly, we have contributed a lot in terms of click chemistry approach, click chemistry, using click chemistry, and so far we have made more than 1,500 new compounds, and all the synthesized compounds are well characterized by means of spectroscopic techniques, well studied their therapeutic properties in terms of anti-tubercular, antifungal, antibacterial, anti-inflammatory, as well as anti-cancer activity, and received excellent publications as well as some kinds of patents as well. Whatever work which I have shown over here, a part of work carried out by Dr. Mubarak Sheikh, Dr. Tejasri Deshmukh, as well as Dr. Satish Akolkar. Their small contribution just I have shown over here, and the remaining students are also contributing to the use of click chemistry for making small molecules and evaluating for their biological activity. Without help of some of the peoples from different types of institutions, without collaborations, this particular work is not possible. That's why I must appreciate our students for their synthetic contribution, as well as the contribution made by the collaborators from different parts of the India, like Dr. Dhiman Sarkar, Dr. Jay Shetty, Dr. Desh Chinde, Dr. Kherkar, Dr. Sandeep Chinde, Dr. Prakash Jha, Dr. Ram. So many people are involved in evaluating such type of biological activity, and without support from from CSRUG as well as DST and from Dr.B.A.M.U.Aurangabad Marathwada University. This particular work is not possible, and without help of students, so many collaborators. And chemistry, I want to say something over here. Just 1 minute I will take. Whatever chemistry I have learned, it is from Shramajyoti Shikshan Prasarak Mandalas, other senior colleagues, and credit goes to all the chemistry teachers, all the physics teachers, all the industrial chemistry teachers, not only these teachers, but also the entire Shramajyoti Shikshan Prasarak Mandalas family, from family, that is other senior college, and I believe on Shrama Yava Jayate. With this, I am very much thankful to my alma mater, that is other senior college. Thank you.
Thank you very much, sir, for your thought-provoking address on click chemistry to set a perfect platform for our international level webinar. In addition to this, you blessed the audience with your valuable words about the theme of the conference, that is the current perspective in chemical science. Friends, now there is the break for 15 minutes to sort out technical problem. We are trying to sort out the problem to increase the limit of the audience. Otherwise, we are going to share the link to join in our WhatsApp groups before 15 minutes. So we are ending the thing, and we will retain the meeting after 15 minutes.
If any queries, questions from audience, just they can raise quickly.
Yes.
Participants, you can raise the queries, questions from your side.
Sir,
Are there?
sir, I have one question.
Yes, sir.
And I must say that it's a great talk, excellent talk, sir. And I'm really so much happy to hear you after a long time. So my question is, what factor decide the regioselectivity of click reaction? And if we wanted to get another regioselective molecule, how can we get?
Yes, it's a great question, and as we are using copper catalyst, which is used to activate the terminal alkyne. Okay, other than copper catalyst, as we are using copper sulfate, copper acetate, copper oxide nanoparticles or forms of the coppers, which regioselectively gives 1,4-disubstituted 1,2,3-triazose. Other than copper, if we use a rhodium-based or ruthenium-based catalyst to carry out click reaction, it leads to the 1,5-disubstituted 1,2,3-triazose. This is a simple answer. To maintain the regioselectivity, you have to have the catalyst, proper catalyst over here.
Does solvent affects polarity of solvent?
I don't think polarity of the solvents, because in different types of solvents, people have tried by using polar solvents specifically, and without solvent also people have performed this particular reaction under microwave-assisted reaction conditions without solvent also. And water is the best solvent to carry out this particular reaction, universal solvent.
Yes, thank you.
Yes, any other peoples?
Sir, there is one question in the chat box. Kindly read that question, sir.
Yeah. I will go through. How much time it takes to screen in vitro activities of such extensive library of triazoles? Lakshman can say, yeah. It totally depends upon the collaborator again, and to grow the strains or slants, it may require a lot of time, and if you start today, it may take a month also, and to have a lot of calculations, and to carry out different types of biological activity, you have to have such kind of collaborations with the expertise from by the biology as well as the pharmacy people. It may take a lot of time. It may take a lot. Yeah. Yeah, there are no questions. Yeah.
Okay, if no questions, we are going to close
I
this.
want to listen Mali sir's voice. Mali sir's
Yes, sir. Yes, sir.
voice. because they are the biology teachers, especially microbiology, and how chemistry peoples can collaborate with the biology peoples. And nowadays, in 21st century, peoples are thinking about the collaborative research, not interdisciplinary, but multidisciplinary approach. Okay, and we could able to have such kind of collaborations with the peoples from not only from India, but also we have collaborated outside the India also. Just a small collaborations we have made, and every students have got trained under them also. These people have not synthesized these compounds, but also they have performed the biological activity part, and it's a difficult part to carry out anti-tubercular activity to handle the TB strain, as well as to handle the antifungal strains of human pathogen like a Cryptococcus neoformans also. And we have observed that some of the peoples, they have lost their lives also while carrying out the biological property like anti-tubercular activity. Okay, yes. Just I want to listen, Mali sir, your voice, sir.
Yes, I have been listening your views regarding the biomolecules and the regarding the chemicals related with the biological molecules or biological system. Very nice. As you are the chemist, scientist in chemistry, but you have studying in the biological molecules related with the
Sir, your voice. Hello.
If they have some questions.
Sir, I could not able to hear you. I could not able to hear you, sir. Hello. Can someone tell me what question Professor Mali sir have raised? I could not able to hear you. Okay, we will talk later. Yes, sir, Mali sir. आवाज येत नाही. Okay. ओके, माळी सरांचा आवाज म्हणजे ऐकू शकलो नाही मी.
हॅलो.
हा, येस सर, येस सर.
येतोय आवाज?
हो हो, येतोय सर. आता आवाज येतोय. येस.
ओके. मोस्ट ऑफ द मायक्रो स्टुडंट्स हॅव attended your session, and they will get definitely benefit of your research and your knowledge. As you are a student of our college, I have told them that all three eminent persons today, they are going to deliver the lectures. They are from our college, and they will get benefit from you in future also.
Thank you, sir. Thank you.
Okay.
Thank you, sir.
Yes. Okay, now we are closing this session, and due to some technical problem, we are going to resolve this within 15 minutes. And afterwards, new link will be provided in WhatsApp group. So all of you are requested to join after 15 minutes, and new 2 persons will deliver their thoughts after 15 minutes break. Okay.
Okay.
To isolate CF18 from the target, we need to, yes, does anybody? So to isolate this F18 from the target, we need to use some carrier like F2 gas. So when this F18 goes into the gas form, it becomes more reactive, and there will be the possibility of side reactions. So we cannot use this electrophilic F18 that easily in the reactions. And in case of nucleophilic F18, we can produce this in cyclotron, as I said previously, by bombarding proton, high beaming proton, and then it makes more less expensive because we are using commercially available O18 containing water to to produce this. And we no need to add any extra carrier like F2 gas I told here. We no need to add any extra carrier, because this F18 will be present in water and it is in liquid form. We can easily handle for the reaction, which gives high yielding reactions. So coming to my actual topic, so we are targeting 5-2A receptor, and for that we are developing a radio tracers which is called as MDL-10907. So the introduction of 5-HT2A, important neurotransmitter in CNS, that is central nervous system. So which expressed in cortical and forebrain region, and contribute to neuropsychiatric disorder like schizophrenia, Alzheimer, and bipolar this and Parkinson disease. So so to detect this disease at early stage, we need some techniques. So we thought to employ these techniques to detect this, and there are no radio tracers still yet available to detect these techniques this disease at early stage. So SPECT and PET can be used to quantify this receptor and measure its occupancy. So PET means positron emission tomography, and SPECT means single photon emission computed tomography. These both are advanced techniques. So in literature, there are some potent radio tracers which shows good activity towards this 5-HT2A receptor. But the problem with this one is this ultrasenin, and the problem with this ultrasenin is it metabolites rapidly, and the resultant metabolites of this ultrasenin penetrate into the BBB. BBB is the blood-brain barrier. It is a barrier which covers our brain and protect our brain from any outside particle. So when this metabolites means when it gets cleavage, the metabolites of this molecule penetrate into the brain. So because of that, it makes more complications to quantify this molecule at that particular site. So that's what this as phase 3 clinical trial, this molecule get eliminated, and then in next generation this MDL-10907 came. So here in this MDL-10907, initially they labeled here 2 methyl group, one at this position and one at this position, as a CLA1 radionuclide. And as I said earlier, this shows very good activity in this is in phase 3 clinical trial. But the problem with this radio tracers is, as I said earlier, this CLA1 has a 20-minute half-life. So this half-life does not match with its biological half-life. Means with this 20 minutes, if you inject this molecule into the animal or in human, so it before reaching its target, before only it get decayed. That's what it makes more complicated to quantify this molecule. So since this molecule has fluorine here, so we thought this fluorine can replace with F18, and we can make more good radio tracer. So some people, they tried to label this molecule at this place, and yes, they got, and but they got very low radiochemical yield, and because of the low radiochemical yield, they unable to compare this F18 radio tracers with CLA1 radio tracers in non-human primitive brain, means in monkey brain. So we thought to improve the yield of this radio tracers, and we designed a new. Before that, these are the some few previous reports where this F18 radio tracer, F18 MDL-10907, they synthesized in multiple steps. This Molhulson, they started from this para-nitroacetophenol. Here, initially they substituted this nitro group using F18 fluoride, potassium fluoride, and then they did alpha bromination here by using bromine in acetic acid, and then later they reduced this keto group using TES. And they got this for fluorophenyl ethyl bromide. And this compound they coupled with pyrrolidine NH, and they got this MDL-10907. But since these are multi-step process, it ended with low radiochemical yield. And second report in 2014, this Ritter and et al. They reported they took nickel complex and then they labeled with TBAF, which is tetrabutyl ammonium fluoride, and they got the similar product here. And then again they coupled with this pyrrolidine NH to get this one. These both are multi-step synthesis. Because of that, it gave less radiochemical yield, which we called as RCY. So in literature, there is a reaction where this pinnacle boron ester they used in the F18 radiofluorination. They reported in 2014. So we thought to employ this reaction in synthesizing this MDL-10907, and we did a retrosynthesis. And we so this fluorination reaction can be used as a late stage radiofluorination reaction, and then this pinnacle boron ester can be synthesized from this key intermediate ketone pyrrolidine. And here this ketone can be synthesized using Lipscomb-Strogel reaction, cross-coupling reaction. It is a known name reaction which developed in Emory University, one of our professor, that we thought to employ in this synthesis. So we began our synthesis from commercially available this compound 1, and initially we did this thioester formation reaction by coupling of acid with thiophenol in presence of DCC and ethyl acetate, which gave compound 2. And then at this point we employed that name reaction, that is Lipscomb-Strogel reaction, cross-coupling reaction between this boronic acid and thioester to give this compound 4. Then this Bock was deprotected using trifluoroacetic acid, which gave this free NH. And then this free NH was coupled with this substituted phenyl ethyl bromide to give this compound 7 and 8. When F is equal to when R is equal to F, it is 7 compound which gave 89% yield, and when R is equal to I, this gives 91% yield. So later, then again we reduced this carbonyl group using sodium borohydride in methanol, which gives this MDL-10907 is 2% yield, and compound 10 in 80% yield. Having this iodo compound in hand, then we actually, this iodo compound is in racemic form, and we need a specific chiral R compound. So at this stage, we separated this chiral racemic compound by making a diastereomer with this chiral moiety, and this chiral acid we coupled with this alcohol in presence of DCC-DMAP, which gives 2 diastereomer, and this diastereomer can be easily separated with column chromatography. And then this required diastereomer we took and we did Mukiyama reaction here to introduce pinnacle boron ester here by using boron pin and palladium DPPF and potassium acetate as a base to give this compound 14. Having this substrate in hand, then at this stage we perform this radiofluorination reaction using potassium fluoride F18 in presence of copper oat reflect pyridine in DMAP at 120 degrees centigrade for just 20 minutes. And then this ester was hydrolyzed using base, that is sodium hydroxide, in 10 minutes, which gave this F18 MDL-10907 in 50% yield. So the previous report they shown only 2 to 3% yield. So we synthesized this compound in 50% radiochemical yield. So having enough quantity of this compound in hand, then this compound we used in imaging of monkey brain and compared with the CA11 images, where you can see these images looks far better and clear than this one. And now after this, we wanted to go for the further step, and we wanted to use for human before going. So, and my next project I'm working on, that is synthesis and evolution of F18 radio tracers for imaging mutant IDH1-2 expression in glioma by PET. So just I will give small introduction for IDH1 and IDH2. What is this? Actually, this IDH is an isoscytate dehydrogenase enzyme, which is responsible to maintain to produce energy in brain. So in human, there are three types of IDH: IDH1, IDH2, and IDH3. So this IDH enzyme, they participate in oxidative decarboxylative reaction to generate energy in the brain. And these IDH enzymes are closely associated with cancer risk. So how they associated with cancer risk, I will tell here. So I am not going to this theoretical part. Just simply I will explain here. What happens is, in brain, this isocitrate is there. So as I said, IDH involves in the oxidative decarboxylation reaction. This isocitrate get oxidative decarboxylation reaction in presence of IDH, and it produces this beta glutarate, which is responsible to produce energy in the brain. And what happens is, when this IDH get mutated, this mutated IDH further reduce this keto group, which gives this 2-hydroxy glutarate, and continuous accumulation of this 2-hydroxy glutarate in the brain, which leads to the formation of tumor. So there is no radio tracers available till now to image or to identify this mutant IDH1 in brain till now. So we thought to develop a radio tracers to image this IDH1 in brain. So these are some potent molecule in literatures. In that, this venasidenib and inacidenib are the drug they used for to treat this mutant IDH1-2. So we selected one potent molecule to develop as a radio tracers, that is this AG881, and then this AG881 contains this chloro group at the adjacent to this nitro group. So we thought to substitute this chloro group with fluorine 18 to produce a radio tracers. And we also, along with this, we also selected another molecule, which is the analog of this AGI12926. And this is a potent molecule for selective for IDH2 and only IDH2. And this is selective for IDH1 and IDH2, dual inhibitor called as. So after that, we started synthesis, and first we followed a reported synthesis, where first we introduced this pyrrolidinone ring into the molecule by using this methyl ester in presence of urea. And then later we introduced 2-chlorine group at this position in triazine. And then this 2-chlorine we substituted with trifluoroisopropylamine to give this AG881. But actually the problem with this synthetic route is, in first step we are getting only 28% yield. And with this lower yield producing, it's very difficult to complete the molecule. And again, another problem is, in second step, we need to use POCl3 as a solvent for this reaction. And that POCl3 we need to remove in the rotavapor. And this POCl3 gives very pungent smell. So while removing, everybody gets irritated. So that's what we thought to avoid this synthetic route and develop our own synthetic route. And then again, then what I did is, I took cyanuric chloride, which is commercially available solid, and that one, the 2-chlorine group was substituted confirmed with HPLC by comparing F19 standard with F18 standard, where we observed the correlation of this both peak, which confirmed the formation of required product. So now these compounds we are using in first cell study, then we go to the rat study, and then now the next analog is this for a mutant IDH2, and it is a highly potent molecule, and it shows nanomolar activity towards IDH2. So this compound has this hydroxy group. So if we display this hydroxy group with the fluorine, it will not affect much more to the biological activity of this compound. So we thought to display this with F18, and we divided this molecule in 3 fragments, and then we started synthesis similarly as I shown previously. First, we introduced this triazinone cyclic triazinone group here, and then we introduced this 2 chlorine group, and then substituted with this 1 amine fragment, and then later we used this propane-aminol to give this hydroxy analog. And then at this stage, we did the tosylation reaction to make a leaving group here, and we perform first fluorination reaction on the bench. So when we perform fluorination reaction, we confirm, we try to confirm function of product by using proton NMR, and there we observed there all the number of protons were matching with the required product. So then we thought to confirm this compound with C13 NMR, and I took C13 NMR of the required product, but this fluorine attached carbon we did not observe. Generally, this fluorine carbon comes at 290 region, and it gives as a doublet because this is magnetically active element, but we did not observe that. So we got doubt maybe something else form, and then I took fluorine NMR of this peak related to that compound. So then we realized that what was happen is this F minus initially, instead of attacking this instead of substituting this OTS, it took proton from this nitrogen, and this nitrogen substituted this OTS, which gives this triazine ring, pyrrolidine ring, 3-member cyclic nitrogen containing ring. So which didn't give our required compound. So what we did to avoid that elimination, side reaction, we thought to synthesize this sulfamidate moiety, and which already used in the literature for the F18 labeling. And since this sulfamidate they will synthesize from 1,2-aminol, our moiety also has, our molecule also has 1,2-aminol. So we thought to synthesize this cyclic sulfamidate using thionyl chloride pyridine and ruthenium chloride as a catalyst, and oxidation with NaO4, which gave this cyclic sulfamidate. And this cyclic sulfamidate we used in the radio labeling of this molecule, which gave this required label radio traces in 11% radiochemical yield. That we confirm with the HPLC, and where we compare this F18 product with the F19 cold standard. Actually, here this red peak is for radioactive detector, and this other peaks is UV detector. So that's what we are using. Generally, we can't take a proton NMR of this F18 standard, because this F18 standard will be in very minute quantity and it is radioactive. We cannot put in the NMR machine. So that's what we need to confirm this with only HPLC. So we successfully, in summary, we successfully synthesized the IDH1 and 2 analogs, and now those analogs we are going to use in vivo in vitro study, and we check binding affinity of these compounds in and tissue distribution, and we take microplate image of these compounds. So I just want to acknowledge my current PI, Dr. Mark Goodman, and our my lab members, and our collaborators, Dr. Hui Mao and Robert Taylor, and all radio pharmacists. They provide us F18 from the cyclotron. And after this, I just wanted to share our some pictures from our group. He is my boss, Dr. Mar Goodman, and here me, and this picture taking my wife here, and this like this our lab meetings goes on here. While lab meetings, we will be having some snacks, cookies, and coffee. Very enjoyable this. And in July we went for a conference, international conference at Canada, Vancouver. There, me and my boss. Yes, and thank you so much for patiently listening me. And if you have any questions, you can ask me. I'm here there to answer you.
If the participants have any questions, kindly put their questions in the chat box.
Yeah, this is quite new area for most of people.
Yes, sir.
Yeah.
I think no question.
Shall we wait for questions, sir?
Yeah, if anybody wants to ask, they can ask me. Think there is no questions.
Okay, I think
Finally, actually, I wanted to thank all my teachers for inviting me and giving chance to present my work here in Adarsh College. And I'm really so happy to present my work in front of all my teachers. And thank you so much.
thank you very much, sir.
PhD
For
abroad. Yeah, somebody asking is how to approach foreign professor for doing PhD abroad. So it's simple. Actually, you need to have good score in TOEFL. You need to give TOEFL exam. It is a English exam, means English questions they will ask you. So if you get good score in that, then then after doing MSc, you need to get some publication during your MSc or after the MSc, and you can approach any of the host here, and you can say them, yes, I qualified TOEFL, and if you guide me, I am ready to do PhD. Then if they get interest in your work, they will take you for PhD.
Thank you, sir.
Yes. Okay. Thank you so much.
We don't have any further questions.
Yes.
So thank you very much, sir, for your valuable address to the audience. Now, may I invite Professor Jabbar Mullah, sir, to introduce Dr. Shivshankar Mane, sir, scientist, NCL Pune. Over to you, Jabbar Mullah, sir.
Good afternoon, everyone. The resource person this session, Dr. Shivshankar Mane, scientist, Polymer Science Engineering Division, National Chemical Laboratory. He received PhD in Polymer Chemistry at Indian Institute of Science Education and Research at Kolkata. He worked as postdoctoral researcher at University of Delaware, USA and Germany. He received prestigious awards, DST Inspire Faculty Award from Department of Science and Technology, he has elected as Young Associate of Maharashtra Academy of Science, and recently he has been recognized as Emerging Investigator by Royal Society of Chemistry at London. His contribution in the field of chemistry, especially in polymer science, is appreciable. Have to stop here. Thank you. Over to Dr. Mote, sir.
Thank you, sir. Now, I invite Dr. Shivshankar Mane, sir, to deliver his address. Over to you, Dr. Shivshankar Mane, sir.
May I audible? Hello. Hello. Hello.
Hello, sir.
Yeah, may I audible?
Hello,
Can you
sir.
hear me? Yeah.
Yes, you are audible, sir,
Okay.
and clearly visible as well. No problem.
Okay, thank you. So good afternoon, everyone. So we gathered here today for this wonderful international webinar, which is organized by Department of Chemistry, Industrial Chemistry, and IQAC. First of all, I would like to congratulate the team who organized this wonderful event. At the same time, I would like to thank all respected teachers and organizing committee for inviting me and giving me an opportunity to share my research views. So it's always a privilege or honor to give a talk in front of the peoples who majorly contribute for your career developments. So I did my master's, bachelor's from others college. So the people, all the respected teachers have major contribution for my research career. So with this, I will start my presentation. Does all slides are visible? Hello? Can you see all the slides?
Yes, sir, we can see slides.
Yeah. So the topic of my today's talk is about the polymers for fundamentals and applications. So why I chose this topic particularly, I kept in my mind that the audience are majorly the graduate students. So that's why I want to give them a flavor for fundamentals of polymers and then some advanced applications. So the basic concept to give a talk for the people who doesn't know more about the polymers. So that's why the fundamental knowledge about the polymer is very important to give the graduate students. So it can open one more window in their addition, what the courses, what they have learned during their graduates. So with respect to the topic about the polymers, as everyone knows, in our daily lives, we use a polymeric materials. In short, I can call it as a polymeric plastic materials. So you all people are very familiar with the daily need uses of the polymeric materials. Particularly, you can see this grandmother having a plastic pot. We can call it in Marathi as a ghagar, or these PET bottles, or these shade nets, and lot of various applications of the polymer plastics in our daily use lives. Particularly when the issue came with the polymer plastics are their like waste materials. People earlier treat this polymer plastics as a waste material. But after the COVID-19, the polymer material gets very high recognition in the field of the chemistry because it become as a solution provider to the society. For example, let me explain how it become polymer plastic become a solution provider. If you consider the COVID-19, before that people feels the polymer as a waste material, but suddenly it come as a solution provider to make the polymeric gloves and shields, masks, safety gloves. So this give a addition solution to the current scenario of the problems. So before going to details about the polymers, let us see what is the background of the polymers or what is the polymers. Polymer is a polymer having a small macromolecular unit joining together. Means poly means many and mer means units. So many units joins together to form a large macromolecule called as a polymer. Here you can consider this as a monomer which joins together to form a large macromolecule called as the polymers. So polymer which is composed from small subunits which is called as a monomers. And the overall process is called as a polymerization process. So earlier days, in a Charles Goodyear, 1844, he introduced the rubber vulcanization process. What is the rubber vulcanization process? The process where he added the sulfur to get the stiffness to the rubber. So he introduced that concept in 1884. Then Backland take it forward for the industrial application purpose. Then in 1920, Hermann Staudinger introduced the concept of macromolecules. So which is for his macromolecular contributions, he received a Nobel Prize in 1953. Then furthermore, he introduced, William Carruthers introduced the father of synthetic polymer chemist. So he take the basic fundamental knowledge of the polymer chemistry to the industrial levels and then further introduced for the daily use applications. So why we choose a polymeric material? What are the key features? So in earlier days, people use wood or metal utensils to use the strength, to give the strength of the materials. But nowadays, polymer overcomes this and use as an alternative to use the wooden materials as well as the metals. It's like an alternative to the metals also. Why it is alternative and why people likes it? What is the key features of these polymers? Because it's an attractive cost effectiveness, which has a very lightweight and high strength, excellent mechanical properties, resistance with respect to the chemical corrosion weather, because you know if you use a metal it will easily get corrode over the time, but polymer doesn't corrode it, and very easy to processable. So polymer can make, using this molding machine, you can mold it with the different kinds of shape using the fundamentals of this processability. Then it's very easy and flexible raw materials and abundantly available from oil industries as well as coal, gas and biomasses. It contains high energy content similar to oil and superior to the wood. That's why people nowadays prefer most of the polymeric materials rather to use for wood and metals. So these are the key features. Then now let us see how polymers are made. Means polymer, as we know that it's a large macromolecule composed from small subunits called monomers. But the process is called as polymerization. If you consider this as a monomer, and when you heat it, the monomer, it forms a homopolymer, means linear chain homopolymers. But when you add another monomer, like two different monomers, which comes together and form a polymer, this kind of structures, like random structures, where you can see the repeating units are not organized properly. So it's called as a random copolymers. And when you add both the monomers sequentially, then you will get a block copolymers. This is a perfect arrangement of the individual blocks. So these are the major polymers made from the synthetic chemist. So with respect to there are sub categories which I will go details. So there are various classifications of the polymers based on the origin, structure. So I will go one by one. So depending on the origin, polymers are classified into natural and synthetic polymers. As the word itself called as a natural polymers isolated from the nature. So it's called as the naturals. The daily life examples for natural polymers are cotton, silk, wool, leather, and cellulose. Or even in our human body, DNA is also a natural polymer. Then another class is a synthetic polymers. The polymers synthesized like a man-made polymers we synthesize in our lab. So we call it as a synthetic polymers. For example, polyethylene, PVC pipes, nylons, terlins. You can see these are the synthetic materials which prepared in our daily life, useful materials in our lab. So the another class of the polymers using a structure. So based on the structures, there are again three class of the polymers. One is a linear polymers, another is a branched polymer, and third one is a cross-linked polymer. So linear polymer, where the structure of the polymers are straight and long chains. Like polyvinyl chloride is largely used for making up the pipes, which is a typical example of linear polymers. And then the wash bottles, particularly what we are using in our chemistry laboratory, even at other colleges as well. So the branched polymer, this is a typical example of the branched polymer. So where linear chains of the polymer forms branches, such polymers is categorized as a branched polymers, which is for typical example is low density polyethylene, or you can say wash bottles. And the third one is a cross-linked polymer. They are composed of bifunctional or trifunctional monomers. It has a strong covalent bond in comparison to the linear polymers, and the degree of branching is very high as compared to the branched polymers, and it gets cross-linked or high-dense materials which cannot be remolded. So that's why it's called as a highly cross-linked polymers. So you can see the typical examples are bakelite and melamines. So this is the switches are made up from the bakelites, and the daily uses dishes, what we are using dishes at our home are made from the melamines. The another class of the polymer is based on the compositions. So like organic, inorganic chemistry, similar way here the concept is as like the organic polymers, the polymer backbone chain is made from the carbon atoms called as organic polymers. All the majority of synthetic polymers are organic polymers. For example, nylon, polyethylene, polyvinyl chloride, and terylene, and many more. So here you can see the daily use applications of these organic polymers. And the another is non-carbon containing polymeric backbones called as the inorganic polymers, where the polymer chain does not contain carbon atoms. So for example, here you can see the PDMS like polydimethyl siloxane, glass, silicon rubbers. And the particular examples of this PDMS is used as a contact lenses in our eyes, and other polymers are daily uses like lipstick and cosmetic additives, which are inorganic polymers. Then next classification is based on the properties. So there are major two, plastic and rubber. In plastic, you can call it as the thermoplastics and thermosets. So what is the thermoplastic? So thermoplastic, the material soften on heating and solidify on cooling. So it termed as a thermoplastic. So it can be reshaped, reused with respect to the temperature. When you use the raw material and give in the molding machine to introduce the heat, it forms a different shapes. So which is soluble in organic solvent, soft, weak, and less brittle. So examples are nylon, polyethylene, and PVC, and you can see more of these atoms. The another class of these plastics are thermosets. So what is the difference between thermoplastic and thermoset? Because thermoset cannot be reshaped. So thermosets are the material does not undergo shape conversion on heating and cooling, it termed as a thermoset. So cannot be reshaped, which is insoluble in organic solvents, hard and strong. So for a typical example of this is our switches, like bakelite switches. And the another class is elastomers. Elastomers are the polymers having viscosity as well as elasticity. So our natural rubber is in this category. So which has a very low cross-link density, high elastic flexibility, and high strength. So these are the typical classification of the polymers based on the origin, structure, properties. So what is the guiding principle? How to choose the monomers, how to design the monomers? Because the monomer should be inexpensive, are easily available, like commercial raw materials are easily available. Biorenewable is very good or preferably materials, and simple reaction condition, very simple way to purify either crystallization or precipitation methods, and easy to go for a scale up stage, and overall yield should be very high. And it's like a typical examples, like versatile for condensation chemistry. So these are particular points to keep in mind to design the polymeric monomers. When we take this monomer to make a different polymerization techniques, how to make a polymers, there are different methods to make the polymers. Typically, it is characterized as like step growth polymers as well as a chain growth polymers. So in step growth polymers, you can see poly addition and poly condensations, and in a chain growth polymers, radical polymerization, coordinative polymerization, and ionic polymerization. So I will just briefly go one by one. So in a step growth polymerization, typically poly addition reactions and poly condensation reactions. So the typical example of this poly addition reaction. So addition of polymer that forms a simple linking of the monomers without any byproducts. So typical example you can see here, this is a polyurethane polymer. The isocyanate and hydroxy react each other without any byproducts to form this polyurethane molecules, which is called as typical example of poly addition reactions. Then another is polycondensation reactions. So in this polycondensation reaction, here there is no byproduct, but here the byproduct is as a water or any alcohol. So condensation polymerization, where monomer reacts with each other to form large structural units while releasing water or alcohol as a byproducts. So this is the typical example of paraphenyl diamine. The next is chain growth polymerization. So in the chain growth, you can see three different ways. Radical polymerization, majorly people use ROM, ATRP, and RAFT. Nowadays, it's very famous. Then coordinative polymerization and ionic polymerization. So what are the radical? Free radical polymerization methods in which the polymer forms successive addition of the radical blocks. So you can make a building blocks of the polymers using a simple monomers. And in anionic and cationic ways, the anionic polymerization, the chain growth polymerization in which active center are ions, either it's an anion or a cations, and it can be considered as an alternative to the free radical polymerizations. So the next is a coordinative polymerization. So you might have studied the Ziegler-Natta catalyst in your chemistry course, where it comes under the coordination polymerization. So in coordination polymerization, the monomers with the side groups are attached through a growing chains. See, these are the particular catalyst. When you take this as a ethene monomers using this complex, it's called as the Ziegler-Natta complex. The Ziegler-Natta catalyst complex forms the polyethylene. And the significant feature of this mechanism is ability to orient each monomer, join it to growing polymer chains in a very specific configuration. And nowadays, people are largely used this Ziegler-Natta catalyst to make these polyethylenes. So what we are using daily polyethylene bags for as a carrier, so which is made up from the ethene to polyethylene using this Ziegler-Natta catalyst. So then I will just briefly go. I don't want to go in details about the mechanism, otherwise you people won't understand in details, but I will just give a brief introduction about the atom transfer radical polymerization. So what is the ATR? It's in short, it's called as a atom transfer radical polymerization. It's a technology to form controlled free radical polymerization, which involves organic halides using a reversible redox process catalyzed reactions. So here you can use as a cuprous halide to initiate the reactions. Particularly, I have given the example ethyl alpha bromoisobutyrate, and this is the styrene as a monomer to make this polystyrene foam. So you people might have known that polystyrene foam as a foam, what you use as a packing material in your laboratory also. We can keep some sophisticated glass atoms inside the plastic foams. Our mobile comes inside the styrene foams also. These the foams are prepared from this chemistry. Then another is the reversible addition fragmentation chain transfer polymerization. So this is the another polymerization techniques which used as a xanthates or thiocarbonates to make the polymers. So particular examples I will show here as a methacrylate or acrylic acids. So most of the acrylates are used as a sheets material using AIBN as a radical initiator and thioesters as a typical raft agent to make these polymers, polymethyl methacrylate and polyacrylic acids. And the another one is a ring opening metathesis polymerization. So it's a kind of metathesis chain growth reactions. So Professor Robert Grubbs invented this chemistry as a metathesis, and for this, his contribution, he has received a Nobel Prize for inventing this Grubbs catalyst generation 192. So what the metathesis process? It's like a changing of the dance partners. If you simply consider this as a two partners, when you introduce the metathesis monomer, it forms and finally it changes the partners. So in this particular case, what we use like stereoregular and monodisperse polymers you can make, and which is simply used from the olefinic cyclobutadiene, cyclopentanes, or narbornine as a precursor materials to make these polymers. And we need a particular catalyst, which is called as a Grubbs catalyst, generation first and generation second. And even now people are using further more generations to make this metathesis polymerization. So these are the typical polymerizations which we use. So once we make the polymers, what is the next step? Then we need to understand or analyze the polymer materials. So for particularly to analyze the polymers, because organic molecules we easily characterize with the NMR, SPLC, and GC as well as mass or IR. But for polymer, we need most important technique, particularly which is developed for only polymer analysis, that is gel permeation chromatography. It is the bread and butter for the polymer chemistry. So then light scattering and group analysis, then dilute solution viscosity and NMR as usual. Then how to analyze the polymer morphology, how the polymer forms self-assembly. So for that also we need to have some morphological techniques. I have listed few: atomic force microscopy, scanning electron microscopy, and transmission electron microscopy. This will give us the information about polymer morphology. So polymer characterization we can get from the GPC, light scattering, N group analysis, or NMRs. But for morphology, we get atomic force microscopy, SEM, and TEM. So I just briefly show you about the gel permeation chromatography. So this is because this is the most important characterization for the polymers and very powerful technique to determine the polymer molecular weight, polymer dispersity. So how we get the polymer molecular weight from these machines? So there is a very simple concept. So I will just go. Before that, I just want to introduce one concept, polydispersity index. So polydispersity index is the molecular weight ratio between weight average molecular weight and number average molecular weight. If the weight average molecular weight and number average molecular weight ratio is 1, then the polymer is called as a monodisperse. Mother Nature provides the polydispersity is 1. Man-made polymer receives the polydispersity index is more than 1. So let us see how the analysis techniques goes and how you can analyze the particular polymers. So I will just show this experimental setup. So here this is the mobile phase solvent which supplies to the delivery, and this is the inject, and this is the injector where you can put your sample and inject to the injector, and then it passes through the column, then it goes to the detector which detects the signals and you can get the data. So this is the machine. Particularly here you can see solvent reservoirs and solvent collectors. This is the sample injection and the columns are here, and these are the UV and RI detectors which can detect the signals. So how the principle goes for that. So let us see. These are the examples of the columns. When you inject the columns, which has a porous packing of microbeads, and each microbead has a pores having a size. So when you inject the polymer, it has a like consider two polymers you injected together. One has a large molecular weight and one is just a small molecular weight. So the large chain need not need to pass and penetrate through each individual pores. So it will go very fast to come out, and the small molecular weight polymers has to pass through each and individual pores. So it takes more time. So that's why the retention time of small molecular weight polymers are very high, as compared to the high molecular weight, because high molecular weight polymers comes as a bypass. It go inside the polymer, so then let's go towards the real applications. So these are the fundamentals of the polymer. And how this fundamental polymer chemistry can take it to the further next level from lab to the application levels. So I will just go for two different examples. One is a polymer for drug delivery application and another is polymer for cellular imaging applications. When we choose as a potential drug carriers, you need the polymer, you need to understand the polymer morphology and polymer hydrophobic hydrophilic balances, which form a different self assemblies. So you can get, because when you varies the hydrophobic hydrophilic balance of this polymer chain, you will get micelles, vesicles, nanocapsules, or as a potential drug carriers. So let me introduce my contribution in the field of the drug delivery. So I have worked on both like covalent approach and non-covalent approach. So in covalent approach, we have used like morning Babu sir already mentioned about the anti-tuberculosis, and even my talk will give some flavor from Lahu Chavan sirs as well. So let us see here. I have introduced the polymer backbone through attached anti-tuberculosis drugs like isoniazid and ribamycin, and which gets cleaved in a particular acidic pHs. In non-covalent approach, where we form the polymeric micelles, in the micelle we can entrap the drug as a, like consider micelle or capsules, where you can encapsulate your drug, and with the external stimuli you can release the drugs. And this is the typical example for anti-tuberculosis drug delivery. So we have used this isoniazid and retinal, because when you take more doses of tuberculosis drug, like a frontline tuberculosis drug, isoniazid or ribamycin, the side effects will come on our eyes. To reduce the side effects, we use retinal as a vitamin A aldehyde, which is very healthy for the eyes. So we introduced this combination and studied about this. So, but I will go details about the non-covalent approach. So in non-covalent approach, how we make the simple polymers and take it for the further advanced applications. We used a simple Diels-Alder chemistry between furan and maleic anhydride to make this norbornene anhydride. Then further it's coupled with aniline and followed by again our basic chemistry Friedel-Crafts acylation reaction using tin tetrachloride as a Lewis acid to functionalize this molecule 3, and finally this coupled with thiourea to get this norbornene derived thiobarbiturate monomer, and this monomer polymerized using Grubbs catalyst second reaction. So we have used here as a ring opening metathesis polymerization technique to make these polymers. Once we make these polymers, the next is to analyze or to characterize these polymers. So we have used as a simple NMR technique to analyze the particular signals of these polymers, where you can see the aromatic proton signals as well as these amide proton signals and olefinic signals are here, which confirm the formation of the polymers. At the same time, we need to check the molecular weight of the polymers. So we have chosen as a gel permeation chromatography, where we injected the samples and we observed the molecular weight is 38,500 with very narrow polydispersity index, and series of the polymers we have made. Once the characterization is over, then what's next? Then to analyze these polymers, so like aggregation behaviors. So to check the aggregation behaviors, typically we need, because at the end we need to introduce this polymeric micelle in our body, so we need to check the size of these materials. Using critical aggregation concentration, we found that it founds 400 microgram per ml to get these aggregates, and having size via dynamic light scattering 96 nanometers with very narrow polydispersity index. These are the typical images of AFM, atomic force microscopy, and this is the transmission electron microscopy. Here you can see this polymer self-assembles in such a way that having a halo in the middle. So you can see typical, this is the hologram, and which has a height size is around 5 nanometers and the width is 96 nanometers. having halo in the middle, like a spherical structures having halo in the middle. And if you see the 3D images of it, looks like a bowl. So these are the sides of the bowl and having halo in the middle, which further analyzed by transmission electron microscopy. So you can see as a bilayer structures having halo in the middle. So once we confirm this aggregation formations, then our aim is to introduce the drug molecule. So particularly we have chosen here as an anti-cancer drug, like a doxorubicin. So we encapsulate the doxorubicin inside these micelles, and using the external stimuli as a pH, the micelle get disrupts and the drug is released, and which we monitored through the fluorescence spectroscopy as well as the UV absorbance spectroscopy. And we have observed that at pH 3, the 78% drug is released, and these are the listed different pH conditions. Then what is next? Once the drug gets cleaved from the body, then what is the fate of the polymer? How the polymer comes out of the body? So to test that, we have tested different biological studies, and the primary biological study is to check the cytotoxicity behavior of the cell growth. So using a trypan blue exclusion method, this study we have done in collaboration with Professor Jaisibda Sharma. And we have seen here, this is just the beer polymer. This is just the doxorubicin. And when we merge, you can see the doxorubicin is in a peripheral of this cell lines. Then what is next? What will happen to the polymer when the drug is released? For that, we have chosen a 8-week-old mice, and we have given this polymer as a gavage dose to the mice and collected urine sample with respect to the time. So because the polymer has a very good property as like a fluorescence. So initially there is no emission, but after certain time it shows the emissions. So we have monitored with respect to the time, like almost 80 hours we have monitored, and the whole polymer needs almost 72 hours to come out the body. So these experiments we have done with the very proper conditions and with the approval of our biological departments. So then what is responsible to form this micelle or micellar aggregates of these polymers? Then, because it's a curiosity for us to check, does this polymer forms only micelle in water or in combination with the different solvent system? So then we have explored different solvent conditions. In a THF, we observed that it forms like a rod-like structures. So which is supported by TEM, AFM images as well as SEM images, where you can see how it forms rod-like structures, because the molecules are arranging each others on a hydrogen bonding induced self assembly, which forms, here you can see typically forms like a rod-like structures, and which is highly compressed to each other to get these hierarchical structures. Then we change the different solvent system, means we just want to change the polarity of the solvents. Then we come with the THF water combinations, 1:1 ratio, and where we observe like a cube-like structures. These are the typical SEM images, TM images, as well as AFM images, where you can see it forms the cubes when you change the polarity. The another typical example is THF methanol. When you change the solvent from THF water to THF methanol, the morphology changes. So here, in combination with THF methanol solvent system, we have observed sphere-like aggregates, which forms spheres. Then we introduced the iron nanoparticles to go for imaging applications of these materials. And then we labeled this and studied how the morphology will change. But we have observed that even after introduction of the iron nanoparticles on the polymer micelles, there is no change in its morphology. So its morphology as a same. Then second is a click chemistry based polymeric nanoparticles for cellular imaging applications. So already morning Babu sir has introduced about the click chemistry, and recent Nobel Prize awardees, Professor Caroline Bartolozzi and medal as well as a Sharpless, who received 2 times Nobel Prize in the history of the chemistry. So what is the click reactions? Already Babu sir given very detailed study about these click reactions, but what is the role of the click reactions in polymers? So let me show in brief. So click reactions, we people as a polymer chemist introduced as to make this linear polymers, cyclic polymers, graft polymers, blockopolymers, or like star-shaped polymers, hyperbranched polymers, or dendrimers. So my contribution in the click chemistry is we have worked on two different ways, like a conjugation with this polymer peptide conjugates or polymer conducting polymer conjugates, and particularly for bioconjugations, polymer and protein conjugations. This work we published in AnguChem, and this is a polymer for cellular imaging applications. I will go in details about this work. This work we published in a small and accepted as a cover image for this journal. So what is the simple chemistry we used, like click chemistry, and what is the benefits? See, if you see here, like a PEG fumarate, this is the one monomer. The another monomer is this tetrazole monomer having again fumarate, and this is PEG melimides. So we have chosen three different monomers, and we made these two polymers. One polymer in combination of these two, which forms this polypyrazole 1. This is the just simple click reaction between this tetrazole and this diene molecule to form this polypyrazole, and